we think that there's a central role in DNA damage and DNA instability, genomic instability, that we want to address.
Hello, everybody. My name is Jose Navarro Betancourt, and I am the scientific director of quadraScope. We invest in early-stage biotechnology companies that aim to dramatically extend health span by targeting the hallmarks of aging. Today, I will have a conversation with Chris Bradley. He is the CEO and co-founder of Matter Bio, a quadraScope portfolio company specializing in diagnosing, preventing, and treating diseases associated with the loss of genetic information, such as cancer. Chris, thank you again for taking the time to do this interview.
Yeah, thanks for having me, Jose.
So Chris, why don't we begin with a quick introduction to Matter Bio? Matter Bio has 3 branches, and I'll let you explain them.
Definitely, yeah. So as you very eloquently said, so Matter Bio is focused on understanding, reversing, and potentially removing cells that have lost their genetic information or information integrity. And we can dig into what that really means. And so that's really our overarching focus. It's understanding what information loss occurs, what it can do, both negatively and if it has any positive effects, reverse it where necessary. And if it's gone too long or things are too far gone, too corrupted, as it were, we aim to remove those cells before they become dangerous to the patients. And that's really our goal.
So genetic information. So for a bit more context, genomic instability is one of the so-called hallmarks of aging. And this means that the mechanisms to repair DNA fail with age. Can you please dive a bit more into what does it mean to say genomic instability? What's information loss?
Yeah, absolutely. So I guess I'll describe it first, and then I think we should address the hallmarks discussion, because there's an interesting corollary to that. So fundamentally, what happens is, as we are alive, every living creature, every living being gets DNA damage. This happens endogenously, so inside the body, inside the cell, or exogenously, so smoking or pollution or things that damage DNA accidentally. Those all damage the DNA. Most of it gets repaired, but occasionally mistakes are incorporated. And what we mean by mistakes is, as if you recall, DNA has multiple letters, A, C, G, T, and each location of those letters is really important. So a mistake is basically the wrong letter in the wrong place. So you really can think of it like if you're reading a book and you see mistakes in each of the words, misplaced letters, that's what's happening in the DNA of each cell as it gets repaired, but missing some of these mistakes. So as over time, these mistakes accumulate, and you can think of that then as noise, right? Random changes that are not anticipated. That's what we mean by loss of information. It's really the information of your genetic sequence that is slowly getting corrupted over time in each cell. And so back to the to the hallmarks of aging. We believe, yes, genomic instability is a core part of that, but we actually are seeing evidence showing that DNA damage and genomic instability is at the core of other hallmarks of aging. So not all hallmarks are made equal. We think that there's a central role in DNA damage and DNA instability, genomic instability, that we want to address.
And Chris, there is increasingly more and more talk about the interplay between physics and biology. Is age-related loss of information related to physical entropy, as in entropy in information theory?
Yeah, so that's a really wonderful way of putting it. You can really think of it that way. So essentially, your DNA is constantly trying to maintain its highly complicated structure. And this, you could consider this an entropy process where randomness and noise and lack of signal starts to enter the sequence of the genome over time. And so nature and evolution has found many ways to solve this. And it's never perfect. So yeah, this is a constant battle that all of our cells are constantly fighting. And I should add, when that battle is lost, then this entropy can turn into disease. And that's really why we care, is it looks like the same process, this entropic process that leads to general aging, is also directly linked to cancer and potentially a whole array of other known illness that's linked to age as well. So there's this very elegant unification happening around this.
And Chris, talking about multidisciplinary fields, you have a very interesting background as a computer scientist. Can you please tell us a bit more about that, and what is the role of computer scientists in life sciences?
Yeah, so it's a great question. So I have a background in both neuroscience and cell biology and computer science, and that was until recently a very non-traditional combination. But since I'd say over almost a decade plus, computational biology and the need for computation in biology has just increased exponentially, primarily because of the amount of data we're getting from the body and from biological systems. So with sequencing technology, proteomics, RNA, seq, all these different approaches to understanding what's going on, they generate massive data. And so you really do need some kind of computational background at some point to make sense of it. So that's part of that. And then also, I think it's really influenced my view of DNA as an information molecule. So really influencing the information theoretic side, which is really computer science, and its convergence around aging. That's been a huge influence for me. And then finally, I also have a background in just tech, and that has, and we could talk about that, but that's really influenced how I can operationalize in biotech and trying to get a positive influence from the tech side.
Yeah, talking about your background in tech, you previously founded Mana Health. What did you learn with this company, with this venture?
Yeah, so Mana Health, my previous company, that was focused on healthcare and health technology, and so it had an element of healthcare, especially around this very highly regulated information in healthcare. So it's a highly regulated industry, but it still had the elements of tech, which is rapid iteration, fail quick, and basically get something out there and shipped as soon as possible to test your theory in the marketplace, which, as you know, is pretty much the opposite of biotech, right? You can't really obviously just hope things work and see what happens. It has to be really rigorously tested. So for me, I learned how to operate a complicated, very fast moving organization in a healthcare environment. And what I'm trying to do with our current company, with Matter Bio, is take the lessons of how do you innovate quickly and test assumptions quickly. They're not going to be tested the same way, right? Biology is not computers, is not tech, right? It's very difficult to predict what will happen until you test it. But I'm really passionate about moving quickly, right? You can still move much faster in biotech than people realize, as long as you're very rigorous about testing your assumptions early and often in a scientific context. So in general, Matter Bio is approaching this problem of longevity and age-related disease as an engineering problem. And that really is fundamentally a similarity to tech, is how do we engineer solutions given the massive amount of information that we now have on these systems, and how do we do that quickly?
So let's dive into these three branches of Matter Bio. Let's begin with Mutagen Tech. If you have to tell us really briefly what's their mission, what's their goal?
Yes. Okay, absolutely. So Mutagen Tech, so this is a sequencing company fundamentally. And the problem it's solving is when you use normal, what we call normal sequencing, it's called next generation sequencing, or NGS. The issue that happens is you basically get 30 copies or more of a particular area of the genome. So you may have heard of 30x sequencing. That's what that is. And for each location, those 30 copies are compared to find the average at a specific location. And this is how we do normal sequencing. So the reason we do that is if you have a mistake somewhere, you'll see a slight change somewhere and you can just ignore it because most of the reads, most of those areas are the same letter. The problem with that is you can't figure out which mistake is a artifact, so a sequencing error, and which is actually a mutation. And the reason for that is many of the mutations we want to study are ultra rare. They're randomly occurring. They may occur in only a single cell. And so basically, they've turned into noise using ordinary methods. So the Vij lab, Professor Jan Vij, who is the director of the genomics department at Albert Einstein College of Medicine, he's one of the premier researchers in somatic mutations. His group realized very early that in order to understand what the role of these mutations are, we need to be able to see them. And so Mutagen tech is spun out of technology they invented to see these mutations. So it's an ultra high resolution sequencing method. And I can share more about how we do that.
And in terms of resolution, in terms of accuracy, how does error corrected next generation sequencing compared to the traditional next generation sequencing, which is already very accurate, over 99% accuracy?
Yeah. So essentially, it's to reimagine it. We are able to detect a mutation that is present in a single cell, in a single location, in one strand of DNA. That's how accurate we are. So in a sense, you cannot get more accurate than this error-corrected methodology in terms of what's visible. And that's how we like to think of it. So another way of saying it for the scientific crowd is in terms of variant allele frequency. So how often a mutation occurs. And most of these techniques require these mutations to be very common. So you'll see, for example, cancer sequencing. All the cancer cells share these mutations, so they're visible. We don't need that. We can see it at a single cell resolution or a single location resolution.
And just to clarify, what can you do in terms of practical things? What can you do with error-corrected next generation sequencing that you cannot do with the other type of sequencing? Like why is it such a big deal to identify this single nucleotide polymorphism or these single point mutations?
Absolutely. So this is where it gets really exciting. So essentially, regular next generation sequencing, like you said, is very accurate, but effectively what it gives you is the original copy of DNA you were born with. That first cell that then became your whole body, your germline DNA, that's what you get when you're sequencing with next generation sequencing. What we unlock is all of the changes that every cell has undergone since that point. So you can think of us as unlocking the change over time that occurs to all these different cells, the mosaicism. So you don't really have one copy of your DNA per cell. You have trillions of different versions. And that area of study, what are those changes and what effect do they have? This technology we have is unlocking that. So it's really, again, thinking of it as what you start with is what NGS gets you, and we're unlocking the change over time, sort of the derivative of the genome, as it were. And that really is completely, it's unlocking a whole new field of study within genetics.
So we're diving into the field of genetic toxicology, basically. And can you maybe give us an example of how these genetic toxicology groups are using your technology? I know you have some big clients.
Yeah, definitely. So yeah, you can think of, so once you can read these changes, then the question is, what's causing them and what are the effects of these changes? And so part of these changes happen naturally, but a lot of them happen because of some compound or mechanical damage. Somehow something is damaging your DNA. So genetic toxicology or genotox is essentially the study of compounds and their effect on the DNA. So an example would be there's some compounds that you probably don't want to be touching or drinking or inhaling. I'll just give a random example. I don't know, brake cleaner, right? If you're a mechanic, we all know that these things are bad, but the reason they're bad is they take your DNA and they damage it in such a way that even though your cell tries to repair it, it can't. And so you have a ton of these new mutations. Now, why do we care? Because one clear result of this is cancer. So what happens is if you get the mutation in the wrong spot, that cell starts to divide and outcompete healthy cells. And if you keep getting these mutations, you essentially effectively end up with cancer. That's the leading hypothesis for how cancer is created. So a lot of our clients are using us because they want to know if a drug or a compound they're using has that risk as early as possible. So back to how do you turn this into an engineering discipline? Well, we don't wait for mice or rodent models or phase 1 clinical trials. We want to know, does this damage DNA in a petri dish? And we can do that with our technique very, very early.
That sounds great. And another example that some people may have heard of in the news, I guess, are the nitrosamine impurities in some drugs, and pharma companies actively monitor these impurities. Will this be relevant for that, for example?
Absolutely. So we are effectively a alternative to the transgenic rodent model that's currently used to study what you just said, these nitrosamine impurities. So for those who don't know, a huge amount of drugs currently on the market, including very, very common ones, have been discovered to include nitrosamines, which is a group of chemicals that certain subgroups of these chemicals are highly mutagenic. So they cause a lot of mutations. So the FDA realized this and has forced companies to prove that their products don't do this. And unfortunately, because of that rush, there's a huge wait list, up to 2 years wait list to actually show the safety of their compounds using traditional assays. And so we are an alternative that actually can show tangibly and quantitatively if a nitrosamine derivative is actually dangerous to the DNA.
That's great. So just to clarify, Mutagen Tech is already operating, right?
Absolutely. Yep. Revenue
Right.
generating and scaling.
That's awesome. Let's move on to another branch of Matter Bio. Let's move on to spell check bio, which now we're moving from the reading part into the repairing part, correct? What can you tell me
Exactly.
about the spell check?
Yeah, so spell check at its fundamental is a editing approach. And I'll explain why we called it that, and it'll be very clear in a second. So what we've discovered already with approaches such as mutagen tech sequencing, error-corrected sequencing, we've discovered that as we age, independent of cause, mutations accumulate in cells, and they tend to peak at around 3,000 to 5,000 per cell at the end of life. And this has actually been shown across a very wide variety of different animals. So mice, dogs, giraffes, whales, humans. There's some really great publications about that. So the problem is we know that this is happening and we know it's a risk for cancer and other comorbidities, but you can't really do anything about it until recently. Now, why? Well, they're random mutations. There's thousands of them, and each cell is different. So if you sequence one cell, you can't use that roadmap for any other cell. Why I bring that up is if you were to treat it with CRISPR or one of the CRISPR derivatives, the next gen CRISPR companies, you can't solve this problem with that. Because if you use CRISPR, you can think of it as a search and replace. Find a sequence of DNA and edit it. And you're going to do that regardless of whether something there is wrong or not. So it's great for diseases that we know very well, like beta thalassemia, for example, or sickle cell anemia. These are huge and honestly groundbreaking cures, potential cures and treatments. But when it comes to reducing noise and overall reversing these mutations generally, you have to have a different approach because CRISPR has off-target effects. It cuts DNA, and so it has a lot of risks associated with it. So spell check is pioneering an editor to find mutations and reverse them only if they're present. So just like the name, imagine you're looking at a Word document, or reading through a paper. We're constantly reading all the DNA, looking for mistakes, for mutations, and then fixing them when we find them. And otherwise, we don't edit anything. And that's fundamentally our approach.
So you have a reader and base editor basically in the single complex. So
Exactly.
so who invented this technology, this CRISPR 3.0, if you will? I know you have very heavy hitters in your in your team?
Absolutely. Yeah. So this was a collaboration and a co-invention between Professor George Church at Harvard, myself, and my co-founder, Sam Sharifi. So the three of us are listed as inventors on the patents for this. And as you said, we really think this is a next generation editor because in order to achieve what I described to reverse these mutations, you have to make it small, you have to make it safe, it has to be repeatable and multiplexable. And so once we show this functioning, we're really showing a much safer and more accurate editing methodology beyond also the use cases that we're looking at.
Let's ground this back into the clinic. Like, why are these point mutations relevant for human disease? Like what kind of diseases can be caused by these random mutations in different types of cells? Like, what are you planning to treat?
Yeah, so the short answer is a massive amount of disease is associated with point mutations and structural variants, which are larger changes. And so we mentioned cancer. Obviously, cancer isn't a single disease; it's a huge group of diseases. So all cancers fundamentally seem to be caused by these point mutations in the wrong spot at the wrong time. So that's one huge category. But another example is what's called clonal hematopoiesis. And what this is a mutation or a set of mutations in your blood stem cells. And that happens as we age. So it's very common. Around 1 in 5 adults over the age 70 have some form of this. And basically, these mutations allow these stem cells to outcompete healthy stem cells. And it has a wide array of issues related to cardiac health. So it increases the risk of a cardiac event, of atherosclerosis, and on top of that is a huge risk factor for leukemia. So that's just an example of what happens when you accumulate these mutations in the wrong tissue. And so basically, yeah, this is relevant clinically to a whole wide array of these types of diseases, in addition to those that you've heard of that are inherited or primarily inherited, like sickle cell anemia and some of these other single point mutation illnesses. I also would like to mention because it's such an open area of study, we've become as a society, we're very good at identifying a disease caused by one mutation, like a beta thalassemia. But when we realized is most disease is polygenic. So there's multiple changes that cause that disease, and our ability to understand those multiple changes is just beginning with technology like mutagen tech. So we already know that we could, in theory, treat cancer and potentially prevent cancer, which would be incredible in and of itself. But we think there's going to be a lot more polygenic disease that we discover through our platforms as well.
And what is these patients with clonal hematopoiesis? What is the care that these patients receive today?
So this is where it's actually very interesting. There really isn't much you can do. So most what are called CHIP mutations, so it's clonal hematopoiesis of indeterminate potential. CHIP mutations are monitored, and occasionally, or unfortunately, some of them progress to what's called myelodysplastic syndrome, which is a indication, considered an indication by the FDA. Not much you can do about that. You basically try to treat the symptoms, but you can't reverse them. And then eventually a percentage of those patients end up getting leukemia, which of course is then treated very aggressively as a cancer. And so we want to prevent the progression that you see there. And we would be the only treatment that's able to do so at this time.
I understand that this technology is still being tested in vitro. When do you expect to move this technology into clinical trials? That seems exciting.
Yeah, so we're very excited about that potential. So it is early, right? These are very much in the earliest stages of research and development. So I don't want to obviously have overpromise on this, but we think within, let's say, the next 5 years, we would be able to get this potentially much sooner. We can get this into a preclinical pathway that then allows us to do phase 1. And one benefit is now the FDA has seen the first gen and second gen CRISPR companies and gotten them through the clinic. And I think there's the first approval recently for treatment in patients with a CRISPR approach. So we are wanting to ride the tailwinds that this provides us, and the regulatory certainty that it's helping bring, to get this as fast as possible into the clinic.
Yeah, definitely. We are now officially in the age of in vivo gene editing. For example, with
Exactly.
therapeutics. Um,
That's right.
Now, let's move on to what happens when cancer already unfortunately happens, and what can you do to treat cancer with Loki therapeutics? How you and how is Loki approaching cancer?
Yeah, so Loki Therapeutics is also some technology out of Albert Einstein, and it's from the Camp lab, Claudia Camp. And what we are able to do is we're able to solve a critical issue in cancer treatment, which is to selectively and safely target cancer cells and deliver payloads to those cells. And the reason that is so difficult is because the ability to differentiate between a cancer cell and a non-cancer cell is fundamental for treatment of cancer, right? We do it through rapid division with chemotherapy, with radiology, with radiology and radiological interventions. You try to focus on an area of identified tumor. What we're able to do is we have a attenuated bacteria. So think of an engineered bacteria that we've designed to only infect cancer cells. And so it doesn't infect healthy tissue, and it only infects cancer cells. And we've designed it to deliver what we want. And so I'll explain what we're delivering next. But that's the fundamental breakthrough: is this delivery methodology to selectively infect cancer. And an added bonus is we found not only does it infect what's called the primary tumor, which is what you usually identify. So stage 1 through 3, you have a primary tumor, which you try to remove. The issue is when you have stage 4, which is actually what usually kills people. It's when cancer metastasizes. It starts to shed cells and set up colonies elsewhere. And this is extremely difficult to treat. And part of it's because it's hard to find those metastases. We've shown that our approach is extremely good at infecting metastases as well as the primary tumor. So it's really a next-gen approach to treating cancer using bacteria as a vehicle. So we've removed what makes normal, or let's say wild type Listeria, so awful and dangerous. That part we've gotten rid of. So it's really not the same creature. It's not the same bacteria. But the reason we use it is it's really good at going from one cell to the cell next to it. And this process is very unique to Listeria, and it's how it gets so into our bodies regularly. So what we've done is we've allowed it to maintain that transfection mechanism, but it only does it in cancer cells. So Listeria effectively colonizes the tumor. That's what we would call it. So you can imagine up to 90+% of a tumor is covered in this Listeria because it's able to go from one cell to the next in the tumor microenvironment.
And in the rest of the body, I guess, is the body able to eliminate these bacteria in healthy cells?
Exactly. Yeah. So it's so attenuated that if you were to give some to a healthy, normal person, they would not have anything at all. So you basically have similar reaction even in cancer patients. In previous trials, this type of Listeria has given them flu symptoms for a few days, similar to if you got a booster COVID shot. You don't feel great, right? Most of us don't feel great, but you don't feel terrible, and in a few days you've resolved. That's similar to what we've seen in previous human populations, which is, as anyone who's unfortunate enough to have dealt with chemotherapy knows, nothing compared to the pain and difficulty of a chemotherapy treatment, which is really what we're comparing against.
So what are you loading this bacteria with? What's the payload that these bacteria transport into the tumor?
Yeah, so this is where it gets, I think, really, really exciting. So Dr. Gravecamp, Claudia, her major insight was, if we want to use your immune system to fight cancer, so immune oncology, right? That's what that's called. Traditional approaches are trying to teach your immune system to recognize tumor cells. That's really the fundamental approach. The problem is tumor cells are very difficult for the immune system to see. It's immune suppressed. And so those traditional approaches don't work very well. What we are doing is we are delivering a tetanus molecule, tetanus antigen, to the tumor. Now, this is the same exact molecule. This antigen is what we give babies to vaccinate them against tetanus. So it's a very safe molecule. We're not worried about that at all. But what we do is we cover the cancer with it. And then all of us who have gotten a tetanus vaccine early in life, your body says, oh, I know what this is. This is tetanus. This is not good at all. Let me kill it. And so, it's a very robust, it's called the memory reaction. It's the purpose of vaccination, right? The reason we do it is the next time your body sees something, it'll get rid of it without you having to worry. That's what we're doing. So we're tricking the body into thinking the cancer is something it's already seen, in this case, tetanus. But we're not limited to tetanus. We could use anything that the body's seen before. So our IP includes things like measles, mumps, diphtheria, tetanus, polio vaccine, anything that's been administered earlier in life, ideally, to the patient.
So what is the stage of this technology, this tetanus toxoid loaded listeria? Have you tested this in animals or?
Yeah, so we have completed all preclinical testing on this. So we've tested it in animals. It's actually been published in Science Translational Medicine. We're actually in the process of publishing a second publication showing it effective in another cancer. So our primary indication is pancreatic cancer, advanced pancreatic cancer, stage 4. We're also showing it as efficacious in stage 4 ovarian cancer. And we think this will work for any solid cancers, which is around 80% of all cancer is some form of solid tumor. So all the ones you can think of, breast, liver, glioblastomas even, et cetera, et cetera. So this is ready to enter the clinic, and we are actually initiating a phase 1/2A clinical trial in the next 18 months as part of our next steps. So this is about to go into patients.
That's amazing. This clinical trial, where will you carry it? Where will you recruit patients?
So right now our lead site is at Albert Einstein. That's our lead site, but we are looking, depending on the phase 1 success, obviously knock on wood, we're looking to expand that. So we're starting at a single site and keeping it very narrow, and again trying to go quickly, fast, as aggressive as we can to get this into patients, and then we plan to expand that to more patients and then more indications as we can. So as I mentioned, we think eventually this has the potential to be a indication-free approach, similar to like Keytruda, where if you have certain properties of a cancer, whether it's pancreatic or ovarian or some other cancer, you'll be able to use this. That's our long-term goal. Yeah, but in the short term, it's at Albert Einstein.
Yeah, the treating solid tumors has always been a problem, and that's without diving too much into the weeds. I think this is basically because of the tumor microenvironment, correct? What makes it so difficult to for the immune system to attack the tumors?
Yeah, so tumors have effectively evolved mechanisms to maintain themselves in a harsh environment like the body, where the immune system is constantly normally on alert to see if something's wrong to get rid of those cells. That's something we do all the time. So what cancer cells do is they end up recruiting other cells in the body that essentially form a shield. And when I say shield, it's not a hard piece of metal or anything, it's chemical. So these cells, they exude chemicals that deactivate and remove the regular immune responses that would normally occur against those cells. That's what we say is the immune suppression of the tumor microenvironment. And this is due to a whole complicated set of co-regulation between the cancer cells and macrophages and other cell types like myeloid-derived suppressor cells. And so, yeah, as our understanding collectively of this has occurred, we've realized we need new techniques to get around this issue, which is a critical issue in this type of cancer.
And yeah, to emphasize this, immuno-oncology drugs are a very big deal right now. Like Keytruda and all the immune checkpoint inhibitors are some of the top selling drugs. And something that's more related to bacteria, you may have heard of T3 pharmaceuticals. They were
Yes.
recently acquired by Boehringer in a deal that is worth around up to $500 million in milestone payments, using bacteria to deliver immune modulators to the tumors. So this is real tech. It's being tested. They're also in
Exactly.
phase 1 clinical trials. So how does Loki's approach differ from that of T3 pharmaceuticals?
Yeah, no, it's a great question. I mean, we love seeing that. Of course, exits are always great for the founders, but what we like is, as much as it sounds very sci-fi, right? It's like, oh, using bacteria to infect and kill tumors. It sounds really out there. It's not. So there's others doing it, and they're being successful, and they're progressing. And so I'd say the main difference between us and them, it really fundamentally is the type of bacteria they're using and the reason they're using it. So they're using it to their type of bacteria, which I forget the scientific name for it, but they're using it to inject proteins into tumor cells. What we're doing is we're infecting tumor cells and then forcing them to express our payload. So similar ends, different means. And then, of course, what we're doing first is this memory recall of the immune system, and that's fundamentally different from what they're doing. But we're really aligned. We think this concept of live bacteria as a treatment methodology is going to be the future. You can almost think of them as little factories, and the implications here are big. So we haven't talked about this, but beyond treatment, this bacteria grows itself, right? So when it comes to scale and being able to treat patients for a wide variety of different indications and at scale, there's a benefit here that's really not encapsulated in traditional approaches where you have to synthesize chemicals, you need precursors. This grows itself. So we think this is really the future.
And you also mentioned that Loki essentially has a platform for any type of payload. What other diseases could be potentially treated with this bacteria as vectors,
Yeah.
for as delivery vectors?
Yeah, definitely. So, so I would say there's two angles of this. So, what can you deliver and where you can deliver it. So, you can deliver now. We've shown this in various publications. Radioactivity, small molecules, CRISPR and other editors, our own antigens, of course, other antigens of interest, and even sensitivity under PET scan. So, basically, being able to track where this goes under a PET scan to see where metastases are if they remain pre and post treatment, regardless of whether the treatment is ours, right? So more of a diagnostic. So we've shown all of this, and so we're really looking to partner with anyone that wants to deliver into the tumor microenvironment. And then, of course, the next question is, how else can you modulate this infection for other cell types and other tissue types? And that for us is the future. So we're focusing, at least in this case, squarely on tumors and tumor tissue because we know it works really well for that. But eventually, I think you'll be able to have tropism for any tissue type, right? It's just a matter of figuring out that chemistry, which is in the R&D funnel, as it were.
That's great. So I believe we covered the three branches of Matter Bio: Mutagen Tech, Spellcheck Bio, and Loki Therapeutics, and they all converge in this concept of genetic information. Is there anything you'd like to add about how these entities are converged within this single holding company within Matter Bio?
Definitely, yeah. So I'd say as a last takeaway, they sound different at first, right? But what unifies them and what we think is going to create, as an entrepreneur and as someone who wants to help people fundamentally with what we do, we think the maximum impact and the most ambitious thing we can do, the moonshot project here, is to really have a unified, vertically integrated solution to the DNA mutation problem. And so as of now, we again, as an entrepreneur, we want to make sure each individual area is de-risked appropriately, right? With clinical trials and with just products out in the market. But if there's one takeaway, it's either we or others and us, we're gonna have to figure out how to solve this issue of DNA mutation. And when you do, you will be able to treat these complicated illnesses, but eventually you'll be able to prevent them. And that's what gets me up in the morning is imagine a day where we can prevent cancer, right? It's almost too science fiction to think about. But now these tools are converging and we think that is in the future. And we want to help bring that about as quickly as we can, whether it's us or someone else. The tools are starting to be there. So it's really exciting.
Very exciting indeed. So Chris, my final question is, what is the best advice you have ever received?
In general, or as a founder, or yeah.
In general, yeah, this is in general. What's the best advice you have received?
I'd say it's a combination of two things. It's one, if you're going to do something, do the absolute best you can at whatever it is you're doing. So 110%. And also don't give up. I mean, honestly, and this is really for entrepreneurship, but in anything, I'd say 90% of my success to date and just the success I've seen, especially in things even like mRNA, right? When you look at the history of the mRNA vaccines and mRNA approaches, it's 30 years of not giving up, of allowing these things the time it takes to show up and work. And so, yeah, if I were to give anyone any advice, the advice I've gotten that's really done well is try your best, truly 100%, and don't give up. And today is going to be different from tomorrow, and you never know, especially in tech and in biotech. Everything can change from day to day. So being around and just continuing to push really is 90% of the battle.
Certainly great advice. So Chris, thank you again for taking the time to talk with me, and best of luck, Chris.
Thank you so much, Jose, for having me. It was great talking.
If you found this video interesting and would like more people to see this kind of content, consider clicking the like button and subscribing to our channel. Thank you for listening.